All right, let's dive right into this explainer and build a rock-solid foundation for understanding the biochemistry of carbohydrates. We are going to map out everything here, from the simplest single monomers all the way up to complex laboratory tests. So if you're prepping for your advanced biology boards like Cambridge A-Level or FSC, well, you're in exactly the right place. We are cutting out all the fluff today and focusing directly on the high-yield structures and processes you absolutely need to know.
Here's our blueprint layout for today. We'll start with life's sweet building blocks. Move into a core comparison of glucose isomers, and then look at connecting sugars via glycosidic bonds. After that, we'll break down the Benedict's test process, make sure we avoid the infamous sucrose exam trap, and then we'll wrap the whole shebang up with a 10-second exam summary. Let's get to it.
Okay, Section 1, Life's Sweet Building Blocks. We really have to start at the absolute foundation, the simplest units that literally make up everything else in carbohydrate chemistry.
So, the crucial point you gotta grasp right away is that monosaccharides are single sugar monomers. I want you to think of them, specifically glucose, as individual Lego bricks. They're the fundamental energy units for respiration, sure, but they are also just waiting to be stacked together to build massive, complex structures. Take a quick look at the general formula right here. C6H12O6. That right there is the standard, everyday blueprint for our most common hexose sugar.
Moving on to Section 2, the core comparison of glucose isomers. Let's see how this all builds up, because even a single Lego brick can come in slightly different, critically important shapes.
Now, focus your attention entirely on carbon 1 in this comparison. Look really closely at these two ring structures. On one side, the hydroxyl, or OH group, is tucked completely below the ring. That's your alpha glucose. But on the other side, it's sitting above the ring. That's beta glucose. Now, this isomer structural shift, it might seem like a tiny, almost insignificant change, but actually it has massive biological consequences for both plant and animal life.
And this breakdown brilliantly illustrates how those tiny little differences dictate completely different polymers. Just look at the contrast here. Alpha glucose chains build your energy stores. So we're talking starch in plants and glycogen in animals. Animals specifically use glycogen because its highly branched structure allows for incredibly rapid energy release, like when a muscle suddenly needs to fire. But beta glucose? It does something entirely different. It builds tough, rigid structural walls, specifically cellulose and plant cell walls. I mean, think about it. You simply cannot build a sturdy structural wall with a soft energy storage brick.
Now listen closely to me here, because this is a massive exam trap. Consider this our high-yield corner for today. Board examiners, especially for Cambridge, absolutely love to test your ability to visually identify and literally draw these specific isomers from scratch. If you're in the exam hall and you accidentally draw the OH on carbon 1 on the wrong side of the ring, you're going to lose the mark. No way around it. So burn that previous visual into your memory right now.
All right, Section 3, connecting sugars with glycosidic bonds. Now that we have our monomers securely defined, let's look at how they actually lock together to form those larger molecules we were just talking about. The mechanism for linking these sugars is what we call a condensation reaction. Think of the glycosidic bond as that satisfying click that locks two Lego bricks firmly together. But, and this is key, when they click, they simultaneously squeeze out a single molecule of water, H2O. If you ever want to break them apart later, you actually have to add that water back in. That reverse process is known as hydrolysis.
Moving right into Section 4, the Benedict's test process. So, the theory is great, but how do we actually detect these sugars when we're standing in a laboratory?
Well, before we test anything, we really need a quick mnemonic. Remember oil rig. Oxidation is loss of electrons and reduction is gain of electrons. This is absolutely crucial because we are testing for what we call reducing sugars. Monosaccharides like glucose have these free aldehyde or ketone groups. That means they have the ability to donate electrons to other compounds, and by donating electrons, they reduce that other compound while becoming oxidized themselves.
So, let's break down the chronological milestones of the Benedict's test. Step 1, you place your sample right into a test tube. Step 2, you add an alkaline solution containing copper 2 sulfate. That's your Benedict's reagent. Now, step 3 is the critical one. You have to heat it in a boiling water bath for about 3 to 5 minutes. Then we move to step 4, where we observe. If you have a reducing sugar present, it's going to donate its electrons to those soluble copper 2 ions, completely reducing them into insoluble copper 1 oxide.
And the results are actually really cool to watch. The more the soluble copper 2 ions are reduced to that insoluble copper 1 oxide, The further along this color spectrum your sample is going to shift. A tiny, tiny amount of sugar, it turns the blue solution green. A bit more makes it yellow, then orange, and a really high concentration gives you this heavy, brick-red precipitate right at the bottom of the tube. This gradient is what makes the Benedict's test such a fantastic semi-quantitative laboratory tool.
All right, Section 5, the sucrose exam trap. This specific exception right here is an absolute favorite for examiners to trip you up on when it comes to the Benedict's test.
So, here's the deal. If you test ordinary table sugar, sucrose, with Benedict's reagent, the test will be totally negative. It's just going to stay blue. But wait, sucrose is a disaccharide made of glucose and fructose, both of which are reducing sugars on their own. So why on earth does their combination suddenly fail the test? You need to know exactly why for your exams.
Okay, the crucial point is hidden right here in the structure. Sucrose features a 1-to-2 glycosidic bond. This specific linkage physically involves the anomeric carbon, which is the reactive electron-donating end of both the glucose and the fructose molecule. Because both of those reactive ends are literally tied up and locked inside the bond, neither molecule has a free aldehyde or ketone group left over to donate electrons to the copper. To actually detect it, you'd first have to boil it with an acid to break that bond apart, and only then could you test it.
And finally, Section 6, our 10-second exam summary. We have covered a lot of dense biochemistry today, from tiny functional groups all the way to lab tests. So let's lock it all in with a rapid-fire revision.
I want you to take a mental screenshot of this summary right now. Seriously, these are the top 4 absolute must-know facts for your biology boards. Number 1. Alpha versus beta glucose differs exclusively at the carbon-1 OH group. Number 2. Glycosidic bonds form via condensation, which means losing water. Number 3. The Benedict's test identifies reducing sugars, changing from blue down to a brick-red precipitate. And number 4, sucrose is non-reducing, specifically due to its 1-to-2 glycosidic bond.
I'll leave you with this thought to ponder as you're reviewing your notes today. If a tiny molecular connection, literally just a few atoms linking together, can mask a sugar like sucrose completely from our standard lab tests, what other biological secrets in the human body are currently hiding in plain sight, just waiting for the right diagnostic test to finally reveal them? Thanks so much for joining me on this explainer today. Keep those notes perfectly organized, and I'll catch you next time.